Good. All right. Good evening, everyone. My name is Noah with Project Lyme, welcoming you tonight to discussing FDA clearance of the Lyme ImmunoBlot test. We have the very well-regarded Dr. Joseph Burrascano, who is a specialist in the diagnosis and treatment of Lyme and associated complex infectious diseases. With over 2 decades of experience and research in the field, he has appeared in and on virtually every form of media, including several Project Lyme webinars, has advised the CDC and the NIH, testified before the U.S. Senate, in Armed Services Joint Subcommittee, and at various governors' councils. He is a founding member of ILADS, who has their annual scientific conference next week, and Project Lyme is a proud sponsor of that. And he is currently an active board member of the ILADS Educational Foundation. So this evening, he is going to be discussing the iDart Lyme IgG ImmunoBlot Kit, which is a standalone test for the detection of IgG antibodies against Borrelia-causing Lyme disease. It features 31 Lyme antigen bands, which are more antigen bands than any other ImmunoBlot test on the market. So this is a very exciting development, and this test has recently received FDA clearance. So I will go ahead and turn it over to Dr. Burrascano to discuss a little bit more in depth about this FDA clearance. He's going to go through his full presentation, and then we'll open it up to Q&As from the audience. So thank you again for joining us, Dr. Burrascano. You have the floor.
Well, thank you very much. It's my pleasure to join you. I'll start sharing the screen. Where am I? Here we go. Do that, and do that, and here we are. Okay. One may think, well, why am I going to give a whole webinar just on a single blood test? Well, this blood test is really the culmination of over 30 years of desire and work by Lyme-related people all around the world who objected to the original CDC-sponsored guidelines for interpretation of Western blot and for serologies in general. Those of you who know about CDC guidelines for interpretation of Western blot, those guidelines have caused a lot of trouble over the years because they originally were for surveillance of disease, but has been misused and quite often intentionally so as a diagnostic criteria. And the problem with that is that the testing by their standards and by their criteria has been very inaccurate. They have a test that doesn't include things that it should include. It does include things it should not when it comes to the chemical makeup of the test. And as a result, there are many, many false negatives and some false positives. Now, people in the Lyme community have known this from the very beginning and have fought to get them to change the criteria. And as of now, the CDC hasn't changed the criteria, but the FDA has gone ahead and approved with full FDA clearance ImmunoBlot that was developed by IGeneX, is now marketed by a company called iDart. And the significance is that this test does include the changes that we have wanted them to make from the very beginning, which at this point is well over 30 years ago. So why has this been such a problem? Why has testing from the big box labs and the hospital systems been so inaccurate? And to understand this, I have to go back to the history a bit. Not only is it a historical perspective, but along the way there have been errors in judgment. Some research has been focused way too narrowly. Of course, things like patents, money, and politics have played into it. So we'll go through this in a little bit of detail. So going back to the 70s, when Lyme was on the radar initially by the people led by Polly Murray in Connecticut, who had a whole neighborhood of people who had diseases involving neurocognitive symptoms, arthritis, facial palsy, the CDC finally sent a researcher to compile the data and do investigations. And that person, Dr. Alan Steer, is an immunologist and a rheumatologist, not someone who's a specialist in infectious disease. He actually thought this was an autoimmune process, as you can imagine. And unfortunately, that bias still exists. And while it's been recognized that Lyme is actually an infectious disease, many, many, many people, including official government agencies, still think that the chronic form of Lyme is autoimmune or at least immune mediated. Well, came along Dr. Willy Bergdorfer from the NIH in the 1980s. He collected ticks from an island off the coast of New York called Shelter Island. And in that tick, he found the Borrelia spirochete that they named it, Borrelia burgdorferi. So that spirochete, which he found in a tick, became what they call the type strain of Lyme disease. And this very specific strain of Borrelia burgdorferi has become the basis, had become and still is the basis of all the CDC, NIH, and university-based research on testing for Lyme disease. And this also became the basis of all the CDC sanctioned and now FDA cleared tests for Lyme until, of course, now. The problem is that if you have a test for Lyme as a lab and you wanted to get it FDA cleared so it could be made into a test kit you can sell, that would have to be as good as what they call a comparator. And their comparator was an ELISA based on the strain B31. It wasn't based on anything better than that. So this is a bad decision in my mind and the mind of many, because here we go. It's likely that a single spirochete derived from a tick and not a human being, came from an isolated island off the coast of New York, does not accurately represent all the variations of Lyme Borrelia that are infecting people all across this country and overseas. So to base the diagnostic testing for all of Lyme disease on this one strain of one tick that came from a spirochete that came from a tick and not even a person, it really doesn't make any sense. Now I'll show you the details on this. It's been found that there are many different species of Borrelia that cause the clinical symptoms of Lyme. And I have a partial list here. There's Borrelia burgdorferi, the strain B31, which is the original strain, the type strain that was found in the tick from Shelter Island by Willie. There's also a human strain, 297, Californiensis, Mayonii, Afzelii, Guerinii, and there are others. So what they've done is they've called this entire group, this inclusive group, Borrelia burgdorferi sensolato, BBSL, okay? So that refers to the whole group of Lyme Borrelia. The one specific one that came from this tick that Willie discovered, that's Borrelia burgdorferi strain B31, and that's referred to as Borrelia burgdorferi sensu stricto, BBSS. So this BBSS, the one I showed in red, is the only species that the large labs use in their testing. And it's the basis until now of all the FDA cleared test kits. But all these other ones are infecting patients in America and around the world and have not been included in any of the CDC or FDA testing, but has been included in the Lyme testing by the IGenics, and especially the ImmunoBlot. And this is the test that now has become FDA cleared. Now, why is it that Lyme has always been called a clinical diagnosis? Well, it's because the testing had been very poor. But unfortunately, when you come with a clinical diagnosis, there's no one sign that proves it's Lyme disease, or if there is, it's not that common. And here's the statistics. Data from the Texas Department of Health and Dutchess County, New York Department of Health show that people with proven Lyme, only 17% recall a tick bite. And people have tabulated those with a rash who recalled a rash or noticed a rash. And from the Texas data, only 36% recalled a rash, and data from other states, 50%. So here we don't have any really solid markers for every single case. I'll show you the data that standard serologic testing, when I say standard, I mean based on the CDC FDA construct until now. These standard tests have now missed up to half the cases of Lyme because they're so insensitive. If you look at cases of early Lyme, if someone presents with a summer flu or body aches and maybe a strange rash that may not be exact for Lyme, the serologic testing at that stage until now has been pretty much useless. And the final paradox is the sicker the patient with Lyme and more ill they are, the more likely they are to have a false negative test. So let's talk about this. The standard serologies are the IFA, the ELISA, the Western blot. They're notoriously insensitive. They only detect that one species, Borrelia burgdorferi stricto, that B31 strain. Now, how these tests are made is also very important. They're made from lysed lab strains. What does that mean? They take a germ like Borrelia burgdorferi, they break it apart, usually by sonication, by sound waves, and they break into all millions of little pieces. They use these pieces, either from the whole organism or some of them, to use in their testing. They use this as the antigens to test and look for the antibodies that the patient may have. To me, this is very imprecise and dirty. You know, there's a trade-off between sensitivity and specificity, because when a whole intact germ, especially a spirochete, is broken apart and all of the ingredients are used to make a test, there are pieces of antigen from this germ that are specific to the Lyme germ, but there are many, many of them that are not specific to Lyme, that are found in other bacteria, that are found even in viruses, and even can cross-react with autoantibodies in people with autoimmune disease. So because of this, there's a trade-off between sensitivity and specificity. In other words, if you have one of these dirty tests, like an ELISA, for example, you determine what's called a cutoff, so that if there's a number above a certain amount, we'll call that positive. If it's below that number, then we call that negative. Well, that cutoff is based on the statistics of how accurate you want the test to be. If you want the test to be more sensitive, you make that cutoff lower, but then the trade-off is that you're going to have a lot less specificity, you get false positives. And if you go the other way to have very, very specific tests, you're going to get false negatives. So when you look at the data, IFA and ELISA, the sensitivity is about 50%, specificity no better than 75%. The Western blot is a little bit better, maybe 50 to 70%, with a specificity of around 80%. Now, to get around this, the only way to really do this with the serology is to use what are called recombinant assays. They result in the highest sensitivity and specificity of any of these tests, and they can be actually engineered in the lab to not only be more accurate for this one Lyme strain, but for multiple species. Now, IGeneX has developed recombinant antigen assays, and they have several of them on the market. They have what's called the LSA, the Lyme Screen Assay, the Broad Coverage Assay, the iDart at-home collection kit, and the ImmunoBlot. Now, this is the data on the Lyme ELISA. Again, most of them are based on lab strain B31, so right away you're missing a lot of other cases. If you look at these studies that were published, the average sensitivity is around 50%. The one here that's highlighted is to show the trade-off that I said about sensitivity and specificity. If you have a test of an average sensitivity about 50% or so, you're doing that to get 100% specificity. You want a very specific test. So if you see a positive, you know it's really Lyme. But that means you're going to miss half. So let's say we turn up the sensitivity to get 3/4 of them. But then the specificity drops and you get almost 20% false positives. And there's no way around this with standard serologies. Now, a Lyme Western blot is kind of the same problem. Again, they take a whole Lyme spirochete, and usually a lab strain. They break into zillions of little pieces, and these antigens that are released are separated out individually, one by one, by a process called electrophoresis. These individual antigens are put on a drop of filter paper. Electric current is put through the bath that it's set in, and these different antigens will spread out on the paper different distances based on their molecular weight and charge. So if you do this and then add patient serum, wherever the patient's antibodies, if patient's antibodies are present, they will be attracted to and bind to these antigens. So what happens is a little dark spot will appear. Now this diagram here, this is an actual Western blot. And see these numbers, these are what they call the bands on a Western blot. They give them a number based on their molecular weight. So here's bands as low as 18 and up to 24 and finally all the way up to the top. So these dots represent places where the patient's serum actually contained these antibodies and bound to the antigen that came from the licensed spirochete. All right, so this is not very good because the way you interpret this is, excuse me, whether or not a band shows up, and if it's present, where on this strip it shows up, what number it corresponds to, and how intense or how dark the spot is. So first of all, excuse me, if you name these bands or identify what protein they're supposed to be based on the number where they ended up after migration, well, that's not very accurate because migration is not a specific accurate thing. The temperature of the bath, the composition of the chemicals in it, all can alter how this looks. Secondly, the lice organism, as I said, releases a lot of non-specific things that are not present in only Lyme disease, but present in a lot of other germs. So you can get bands that show up that look like they're from Lyme, but could be from something else. The intensity, how dark they are or how light they are, actually depends on the strength and quality of the culture that was done to make the spirochetes in the first place. Strong culture gives you dark dots. A weak culture gives you weak dots. Again, that's very non-specific. And overall, that's why the sensitivity is 50 to 70%. And finally, and most importantly, this is only one species. You'd have to do this for every single species of Lyme Borrelia out there, and that's just not done by the labs. Now, could this be why there's such a high rate of seronegativity, why so many people get a false negative? Well, turns out none of the commercial FDA-approved test kits or the big lab Lyme testing, including IFAs, ELISAs, Western blots, or the PCRs, or even the T-cell tests, have been validated for all the Borrelia. And again, I put the same list back up just to refresh you. They're validated for this one species, Borrelia-causing stricto B31, which again came from a tick and not a person. You can't even test for these other ones if you want to use a big commercial lab. So IGeneX then came along and they developed testing that will look for all these and more. And actually their testing is not based on, just lab strains gotten from some commercial source. They actually source a lot of their specimens for testing from actual Lyme patients. Now here comes the CDC, what I was talking about before. They held a conference back in the early 90s called the Dearborn Conference. And this was designed to, or set up to, give criteria on how to interpret that Western blot, what I showed you with all the bands. So the bands are given numbers, again, based on where they migrate. And they named 10 specific bands on the blot. These are the numbers 18, 23, and 0, and so forth. And out of the 10, to be positive, the IgG Western blot has to have at least 5 of these 10. Now the problem is, a lot of patients who really have Lyme don't have 5 bands. Does it mean if they have 4 out of 5, they don't have Lyme? Well, this is their black and white thinking. On IgM, the same thing. They give you specific bands. They want 2 out of these 3. Now, besides being kind of arbitrary 5 out of 10, they included a number of bands which are not specific to Lyme. For example, 18 is a flagella band. Any organism with flagella might have that. Same as band 41. This is present in syphilis, it's present in oral spirochetes, it's present in leptospirosis. And a lot of these others are just non-specific. 66 will cross-react to some viruses. On the other hand, bands such as 31 and 34, which are very specific for Lyme, are not included in their list. And why did they do that? Well, this is in the early 90s when there's a lot of commercial interest in developing Lyme vaccines. And the Lyme vaccines of the day, the one that actually came to market, was a vaccine where they actually vaccinated you with this 31 kilodalton antigen from the spirochete. They actually gave you a spirochete protein. Others didn't, were researching using band 34. So the thinking at the time was, well, let's exclude them from the Western blot. So if someone got a vaccine and then later got Lyme disease, we wouldn't confuse them by saying, oh, these 2 bands are present and you'll know if it's vaccine or not. Well, it turns out the vaccine was a failure. And people who did get the vaccines, the antigens that were resulted actually polymerized. And when you got band 31 from a vaccine, you also got a lot of these other higher molecular bands. So the whole theory behind this was just not valid. This conference was very controversial. Several participants openly disagreed and argued, and one even actually walked out and refused to put his name on the publication. So again, the result of all this is very low accuracy. You have things here that make the Western blot interpretation insensitive and also not specific. Then they came along with what's called 2-tier testing. The idea behind 2-tier testing, when you have a severe illness like this, is to begin the screening with a very sensitive test, very sensitive to the point even of like overreading. We might get some false positives. So then what they do is, knowing that you might have some false positives, they require that every positive test be confirmed with a second test that's just as sensitive, but at least 95% specific, thinking this will then weed out the false positives, okay? What did they do? They made the Lyme ELISA as tier 1, and now we know it's not 95 to 99% sensitive, it's 50%. So off the bat, you're gonna miss half the cases, okay? And that would never be acceptable in any other disease to miss half the cases and say, well, that's okay. So let's say you were positive. So then the second tier would be the Western blot, which, as I showed you, is also not very accurate. They can be false positives and negatives. And because of false negatives, people who even were positive on the first tier could be negative on the second. Anyway, the whole thing turns out to be a very big fiasco because so many cases were missed. They tried to refine these 2-tier scheme by having a second ELISA instead of a second Western blot. That was done simply because the Western blot is labor intensive and the second ELISA can be run on a machine, saves the lab money. Well, the second ELISA was no better than the first, so it's certainly no better than the Western blot. And so even the modified 2-tier test based on this was really not that good. What does this result in? Well, what we call Lyme denialism. Many Lyme patients are told they don't have Lyme, even though we know a negative test can never rule it out. They're given wrong diagnoses, they're told live with your symptoms, even blamed for their illness and say they need psychiatric help. And even worse than that, if you can think of it, even if you were given a diagnosis of Lyme, if you don't have a positive test, insurance companies may deny covering treatment for it. And there's a saying in the industry that the most dangerous test for Lyme is a false negative. So here comes the Lyme ImmunoBlot that IGeneX created. First of all, it's very, very sensitive. Second of all, it has the ability to detect not that one species from a tick, but multiple species, the ones that account for the major or bulk, the majority of symptoms and cases of Lyme in America. Not only that, this is a test that has what I call game-changing sensitivity in early Lyme disease. In early Lyme, it's agreed that you don't even do a serology because it takes 4 to 6 weeks to develop antibodies. Well, it turns out the IGeneX ImmunoBlot is so sensitive that the antibodies that have developed, and they do start developing early, but they just couldn't be detected before now, they actually picked up over 83%, 84% of the cases of early Lyme in this one series. Another series showed even a higher percentage. So finally, we have a test you can use in early Lyme disease. In disseminated Lyme, validation studies have shown sensitivity was 100% of those samples tested, and the specificity also 100%. So again, you see there's no longer a trade-off between sensitivity and specificity. Totally game-changing. Now, why is it so accurate? Well, they don't take a germ and break it into millions of little pieces and try and make some kind of sense out of that mess. They use what are called recombinant antigens. They're created in the lab. They're very highly specific. Once these are created, it's not one antigen, it's a whole bunch of them. As Noah has said, the ImmunoBlot uses actually 31, and more than that, they even have some negative controls. So all of these antigens are now printed on the strip, so they don't use this vague electrophoresis to spread things out in a water bath. They actually use a printer like an inkjet to print these bands on the test strip. And that means they're in exactly the right location. So if you get a band at a certain spot, you know what it is. It's not one of these vague smears. Secondly, because it's printed, you know how much you put on the paper, the intensity of the band is not dependent on a culture that may be good or bad. Now, this is available for Lyme, relapsing fever, Borrelia, Babesia, and Bartonella. It can detect all the major Lyme Borrelia species infecting American patients. In relapsing fever, again, all the major pathogenic species. In Bartonella Babesia, it can also detect multiple species and name the major ones. And as I said, if you're doing a serology or a Western blot, you'd have to have individual tests for every one of these other species. And that's just something that's not done. So let's just compare the two. Here's again that same picture of the Western blot, and it's made from lice cultured lab strains. Now this lane all there in the end, this is the control strip. This is what actually got released in this culture. How would you ever know what's in there? I could never interpret that, even with a microscope. And again, it's this one species, B31 lab strain. The ImmunoBlot, again, look at the difference. Look how clean that is versus the Western blot. And it does include all these other species and even more. Now, I said they're made of recombinant antigens. Well, what actually is that? As I said before, in a standard serology, the test antigens are derived from a whole organism that they break apart. And you can make the test out of the whole lysate, like all the different antigens, like I showed you on the Western blot, or with some ELISAs and IFAs, so pick and choose some of them, and in some cases, just one of them. So if you use a whole lysate where all these proteins are released, you can have cross-reactivity to other organisms and even autoantibodies, resulting in poor specificity. If you use a single antigen, like is done with the Miyamoto test that is offered by the commercial labs, it's one antigen. It's like one band on a Western blot. Not only is that poorly sensitive, it's also very poorly specific because there are other things that have that band. And again, the trade-off is sensitivity and specificity. On the other hand, with recombinant antigens, what are they? What they do is they clone the genetic sequence of the antigens that they want to test for, and they clone them and put them into viral vectors, which are then infecting E. coli. The E. coli produce very highly pure protein, which are then further purified. So what that means is you're getting antigens that are very highly specific to whichever organism you want to test, because you're actually using the genetic sequence of that organism and of that protein to make them. So this virtually eliminates false positives and false negatives. And because you're selecting which one and ones you want to use, you can actually look for a very broad range of species, not just a single lab strain. And again, no longer the same trade-off of sensitivity and specificity. Another thing that happens, a lot of you know this, and as someone who has established long-standing Lyme, and you do a serologic test, you find a negative IgG but a positive IgM. And so what is that? In most infections, IgM, as you know, is an early responder. You see IgM appear pretty quickly, and then in most infections, it disappears after 4 to 6 weeks. However, it's not unusual to see a persistently positive IgM in chronic Lyme disease. There are a number of different validated reasons for that. Anyway, with standard serologies like the ELISA IFA Western blot, this could be a false positive due to cross-reactivity with other organisms, including viruses, because with these serologies, the IgM is not very specific. And this is why the CDC, the IDSA, the NIH always tell us to ignore a late positive IgM in disseminated Lyme. In fact, any IgM positive after 6 weeks, they tend to discount. However, with IGeneX, because it's recombinant, again, you don't have that trade-off of sensitivity and specificity. Because it's recombinant, a positive IgM is not a false positive, it's a true positive. Here's the data on that. The specificity of an IgM on ImmunoBlot is 99.3%. In other words, more than 99% of the time, a positive IgM, even in a late case, is a true positive. Again, this is a game changer because so many Lyme patients, especially the very sick ones, never develop a positive IgG until they're treated. And now you have a test that'll pick it up with a very high degree of accuracy. So, I mean, the bottom line here is if you test someone with IGeneX and they have a positive IgM despite being a disseminated case of Lyme, or any of the IGeneX testing that uses recombinant technology, do not dismiss a late positive IgM. It's a real positive. All right, so you can say, well, this is a lot of lab mumbo jumbo. Does it really matter? I'll give you some case histories. All right, here's a series of patients, and all of these were from offices of different physicians around the country who use IGeneX for testing. It wasn't a directed sample, it was just randomly picked from these doctors' offices. So the first series includes 36 patients from North America who had a negative Lyme Western blot. They were tested by the Lyme ImmunoBlot, and guess what they found? Of these 36, they found Mayonii, Spilmanii, Californiensis, all these other ones, even found strain B31, the one that the Western blot's supposed to pick up, it didn't even pick up. And of all these, they found these that the Western blot did not show, and actually found 5 other cases where they couldn't identify the species. Now, what does that mean? Sometimes you get an ImmunoBlot and you find the result says Borrelia species found or Bartonella species found. Well, if they can't speciate a result, they looked into that, and usually that means that antibodies from multiple species are present. In other words, you could be co-infected with, say, Lyme Borrelia from Mayonii and maybe Afzelii. And what happens is you get so many bands on the ImmunoBlot that it's very hard to pick out each individual species. But the bottom line here, though, is that if you get a positive result that's not speciated, it's still a positive result. Don't ignore it. Here's a second case. 29 patients from American Medical Office. All these 29 had negative Lyme Western blots. But guess what? Again, found Spilmanii, Californiensis, Afzelii, Mayonii. Here again, 2 strains of B31 that should have been picked up but wasn't. Shows you again how more sensitive the ImmunoBlot is. 11 were unspeciated from America and 4 unspeciated European Borrelia. Now, people travel, they fly on jets, sail on boats, and they cross country lines, and they can pick up other infections. On the other hand, birds fly, animals and pets travel too, and there are some European species in American patients, and this proves it. Final series, just I want to, overdo this here. Again, 11 patients, negative Lyme Western blot, and if you add up the numbers here, there are more positives than there are cases. And why is that? Well, this demonstrated that one patient had antibodies to 2 different Lyme Borrelia, another one had B31, and another different Lyme Borrelia. So this is a perfect demonstration that co-infection with multiple species exists and can be detected and proven here on the ImmunoBlot. Now, this is the nitty-gritty on how you interpret the ImmunoBlot and what makes it so special. Unlike the CDC criteria for Western blot, the criteria for reading a Lyme ImmunoBlot is you only need 2 bands, or specifically 2 bands from the individual groups. So for example, band 23 and band 31, or band 39 and 93. But when you look at this, there's not one band 23 or one band 31. They actually have 9 different bans for 23, 9 for 31, 2 for 39, 2 for 41, 2 different controls. As I said, 31 pathogen bans and 2 more controls on that. And why do they do this? Well, they do this because different species have different 23s, different species have different 31s, different 39s. So by using not one band 31 or one band 39 and so forth, by using all the most important ones, this gets them to be much more sensitive, much more capable of picking out all these other species, and why it's so much more accurate and better test. So now comes the FDA clearance. Now that's why this is such a big deal. Okay, because this is the first test since the early days, since the very beginning of Lyme in this country, where we have a test that includes bands 31 and 34, which none of the other FDA cleared Lyme tests ever do. It also includes ability to detect a positive with just 2 band groups, not 5 out of 10. So that is huge. This is what we've been fighting for over 30 years. Now, the other interesting thing, let me go back a slide. I don't know if you can see it on the bottom. They have on here a band for the LSA test, the Lyme screen. The LSA test, the Lyme screen antibody, is considered tier 1. The ImmunoBlot itself is considered tier 2. So going back to this, here we go. This ImmunoBlot that's been cleared by the FDA is actually a 2-tiered IgG test. Okay, so here we go. It's a modified 2-tier test. It's sanctioned by the FDA. It uses the IgG criteria, the 2-band or 2-band group criteria, not the CDC 5 out of 10 criteria. It doesn't include the crazy bands that are non-specific, but it does include band 31 and 34. This has completely cleaned up the problem that we've had for over 3 decades with this government-sanctioned testing. And to get this done, IgG got a great deal of input and help from the Bay Area Lyme Foundation. Even the CDC was very helpful in getting this together for them to get the clearance. Also, so many practitioners donated samples, so many patients donated samples that IgG is just very appreciative of all that everyone has done for them. Okay, so what does this FDA clearance mean? It's important because, most commercial labs and most commercial, in fact, almost all hospital labs, they don't develop their own tests. They don't, grow spirochetes in some corner of a lab and make incubators and all that. They purchase test kits, just like they purchase a test kit to look for thyroid antibodies or whatever. They don't do their own, they purchase test kits. And by law, all commercial test kits have to be licensed, and that means FDA clearance. FDA clearance is a licensing procedure. So now that the FDA has cleared the IgG Lyme ImmunoBlot, which is again marketed as the iDart ImmunoBlot, it means that this ImmunoBlot kit can now be sold to other labs to use. So your local hospital can now elect not to buy a kit from who knows what, but actually to buy the kit from iDart, from IgG, and get this high quality ImmunoBlot as part of their routine Lyme testing. It also means that the big box labs, whether they want to use an ImmunoBlot all the time or they want to use it as a higher quality test to supplement what they already offer, they have the option to do that now. This FDA clearance is recent. The test kits are just now being produced, so I'm not sure how many labs have adopted this yet. You'll have to stay tuned. One other thing, though, to let you know, this clearance is for the IgG ImmunoBlot. The IgM ImmunoBlot clearance has been submitted and is pending. So will this mean that the ImmunoBlots are now going to be covered by insurance? That's not very clear at this point, because I looked this up and there's actually no rule on this. It's up to the individual insurance companies to decide on coverage. And often there's a discussion, let me be polite and say a discussion, between the laboratories and the insurance companies to get their tests covered by insurance. And no doubt there'll be a lot of discussion on this, because how can an insurance company say the test is not appropriate when in fact the FDA itself has cleared it? And again, if a lot of big labs adopt this, and a lot of hospitals adopt this, they're going to pressure insurance to cover them. And just so you know, the ImmunoBlots already are covered by Medicare. We're talking about commercial labs. Now, I did want to spend a little bit of time talking about the AccuDart, because that's another thing that just came out for people to know about. This is an accurate at-home or in-office blood test kit that's available for the major tick-borne diseases. It's a situation where you can get the kit online. It's mailed to you. You do not need a doctor's prescription to get this kit. And you stick your finger, you put the drop of blood on the test strip, and you mail it back to IgG where it's processed. Now, the beauty of this is that you can get this for Lyme disease, tick-borne relapsing fever, Babesia, and Bartonella, either as an individual test, or you can actually get all of them. They call it a tick-borne disease test panel, which contains, at this point, 19 different markers across the 4 diseases. And again, it's based on their recombinant technology. It's an outgrowth of their broad coverage assay. So it's recombinant technology. It's multi-species. The answer comes back as simple yes, no. I'll show you actually a picture of the slip. It doesn't name species or separately report IgM and IgG. It doesn't give a titer. If you need that kind of information, then you would have to go to an ImmunoBlot. Studies have shown a sensitivity of this of 90%. The only test with the highest sensitivity of this than the AccuDot at this point is the ImmunoBlot itself from IGeneX. You can get it online from accu.health.com. You do not need a doctor's prescription. Again, it's a simple finger stick. You don't have to go to a lab to get blood drawn and have it sent off. So this is good because if you're a patient that's new to an office and you are waiting to get your appointment, you can get the blood testing done ahead of time. If you need testing between visits, you don't have to leave your house. You don't have to go to a lab or the office. You can get it done at home. If you are in treatment and want to have testing done every few months to see how it's evolving, now it's easy to do. Also, if you're having symptoms recurring or something changing, maybe after treatment has ended, you want to see where you stand, you can also get the testing at that point. Another thing, very often, family members, once you've had Lyme, you start looking at family members, say, I wonder if you have Lyme. Let me get you tested. And they say, nah, don't bother me. I don't want to see the doctor. And, you've heard the stories. Well, this time, I said, look, give me a finger. We're going to stick it. We're going to get an answer right now. So this is a way to very easily screen family members and loved ones who otherwise would not want to go to the trouble of going to a Lyme-related doctor and spending the time and all that. And again, if you want to expand on the results, in other words, this is a simple yes or no. If you want to expand, see what species, is it IgM, is it IgG, is it both? You can always get an ImmunoBlot afterwards, but you don't necessarily have to because this is not a screening test. This is a bona fide ELISA replacement. Okay, I want to just summarize the different tests that are available because I do want to field the questions. I want to leave time for that. These are the ones. These are all recombinant technology, okay, the serologies. So we'll start with the LSA, the Lyme Screen Immunoassay. It replaces the IFA as tier 1 in a 2-tiered test. Here the ImmunoBlot is tier 2. The FDA cleared Lyme IgG ImmunoBlot from IGeneX includes the LSA, so when you get that test done, you're getting the 2-tier test. The broad coverage assay and the AccuDarts, they're replacement for the ELISA. Result is, a positive or negative. It's a very simple, clean test, easy to do. Does not report separately IgM or IgG or give a titer. Doesn't tell the species. It just says yes or no. So it's an accurate test, but very simplified and pared down, and that's why it's less expensive. ImmunoBlot, that's the gold standard, the Cadillac of testing. It's the most accurate and comprehensive serology available. It does cover all the different species. Again, it's recombinant. It reports the bands, it reports IgM and IgG separately, will name most of the species. And if it can't speciate it, it'll tell you that, which might give you a hint that either you have an atypical species that hasn't specified or hasn't identified the bands, or it could mean that you're co-infected with more than one Borrelia organism, or Bartonella or Babesia. So that's an important clinical clue, and I think that's a big reason why ImmunoBlot should be ordered. IGeneX also offers a T-cell response assay. T-cell responses are independent of B-cell responses. In other words, you can have a positive T-cell test even if you're seronegative. And of course, they also offer the culture. The culture is an advanced direct test where the blood is actually incubated for a couple of weeks in a laboratory incubator. And at the end of the 2 weeks, it's analyzed, and the positive organisms that are found are then confirmed and identified by a very highly specific and very sensitive PCR. At this level of culture, what they're doing is they're reporting at a genus level. This way gives you maximum species coverage. So, for example, they'll find a positive Borrelia that would cover all the major Borrelia species, or positive Babesia, all the major Babesia species. And in fact, as many of you might have heard from my other presentations, they actually found some very strange and atypical species that were not supposed to be infecting human beings, like dog species and pond species. But they actually have found these because it's such a highly sensitive test, and has actually cleared up a lot of mystery illnesses already for some patients. So I'm going to stop at this point. I'll give the screen back to the host so we can start going through, how we do this, here we go. So we can start going through some of the Q&A.
Awesome. Thank you so much, Dr. Burrascano. I think that was a great presentation. As we discussed beforehand, I expect one or two of these Q&As might address information that you've already gone through, but obviously, can be very dense. So hopefully we can just briefly touch on one or two of those topics again.
Sure.
I briefly wanted to respond to some housekeeping questions that I had seen. A couple people asked if this was being recorded. Yes, it is being recorded. And if you registered, which you obviously did since you're here, you'll receive a link to the video first thing tomorrow morning. I also saw a question about one of our prior webinars. The link that I just dropped in the chat is all of our recorded events that you can go to our website and rewatch for free, 100% on your own timeline. So we do record all of our events and make sure to post them afterwards. So that is a clear yes, and very excited to hear that people are interested in seeing past webinars that we've hosted. Now to get into the topic specific questions, I'll leave about 15 minutes for this, and hopefully we can get through as many as we possibly can.
Okay.
The first one that came up here, and I've seen this question a couple of variations, is this test viable for other tick-borne diseases? And I know you did just briefly mention that in your last slide, but I think it might be helpful just to directly answer that again.
The Lyme IgG ImmunoBlot is the one immunoblot that's now FDA cleared. They have an IgM Lyme ImmunoBlot, which is going for clearance, has not been through all the paperwork yet. But they do offer immunoblots for other diseases. Those have not yet been submitted. They're doing one at a time. So stay tuned.
Awesome. Yeah, hopefully those get approved as well. That would be really, I think, exciting for patients across the sphere.
And one thing I have to say is that from what I've heard, the government agencies have been very, very cooperative because they have worked with IGeneX as partners, not as adversaries, and they developed a kind of a good working relationship. And I think they'd be happy to help them get through the next phase of approvals for the other pathogens. I mean, I'm just saying that. I don't know. I don't work for IGeneX. I don't know the internal workings, but I know it's been a very congenial relationship, and I would not be surprised if the other ones get approved as well.
Yeah, definitely jointly beneficial, just given the fact that there's so many patients out there that are looking for answers. So if there's something that is effective and clearly has a background of having worked, then I would hope that it gets FDA cleared as soon as possible. So one other question that I've seen come up a lot is about the effectiveness for chronic patients or post-Lyme syndrome patients. It's called a variety of different names, but are these tests, specifically the one that was recently FDA approved, aimed at patients who are newly ill, or does it apply across anybody who might have tick-borne disease?
Well, they've done studies on all stages of Lyme with the Lyme ImmunoBlot, for example, and they found it is the most accurate serologic test for all stages, everything from the earliest pre-rash stage all the way up to the chronic and disseminated cases. One of the problems with chronic Lyme, as a lot of people know, is that it's a disease that impairs the immune response. Serologic tests, including immunoblots, depend on antibodies being present that they can detect. The reason why the ImmunoBlot is so successful is because it can detect even tiny amounts. That's why it can pick up early disease and late disease. But still, there are some cases where either because the immune system is so deficient, it's just not making the antibodies and enough amounts to be detected, or there's another paradox. People who are very, very ill have a lot of germs, a lot of bacteria, a lot of spirochetes in their circulation, and the antibodies bind to them. And that's how antibodies work. They bind to the pathogen, and the rest of the immune system recognizes that and starts to try and eliminate them. But if you have so many organisms, it binds up all the antibody that your body can make, and there's nothing left to be picked up by the test. In other words, the test will pick up antibodies that are freely circulating, not those that are bound up to the organisms. They call this binding immune complexes. So that's the reason why there's this paradox in late Lyme, either because of immune deficiency or these immune complexes or both, you can get a false negative test. And that's also why some practitioners will treat patients and then redo the serology, redo the ImmunoBlot, and sure enough, it becomes positive. I actually did a study on this, and the conversion rate was at least 36% and sometimes even higher. But that's one of the reasons why people are giving some treatment and then redoing the serologies.
Thank you very much. I think that kind of covers the 3 or 4 varieties of that test or that question that I've seen put in the Q&A here. There's a variety of people asking about, chronic Lyme or disseminated Lyme or post-infection. How does it separate, etc. And I think you did address all of those issues.
Also, don't forget, as I mentioned in the presentation, a lot of these people don't have an IgG response, only IgM. And the ImmunoBlot will pick up the IgM. Now, it's not yet FDA cleared, but the standard ImmunoBlot is available from IGeneX, has been since, 2017, I think. So it doesn't mean you're not going to be detected. In fact, you will be detected in much higher sensitivity and much higher specificity than any of the other tests out there.
Yeah, that was a perfect restatement. And actually, one of the other follow-up questions that I had here was asking for the distinguishing between the IgG and the IgM. Just to clarify, the IgG is the one that was FDA approved. All of the other tests are still offered by IGeneX at this time. However, the IgG was the only one that was FDA approved. So just keep that in mind, everyone. And I definitely recommend rewatching this presentation afterwards. Again, it's being recorded, and it should allow you to go back in and listen again and a little bit deeper to some of the explanations of that, because I understand it can definitely get complicated with the
maybe you can slow down the recording.
Yeah, definitely a wide array of tests offered by IGeneX. And again, the hope is that more do become FDA approved, but it's just the IgG at this time. So this question is about blood draws. It's any idea whether the blood can be drawn via a port or a regular draw instead of the dart? And would that work? And is that a part of the FDA approved test?
Well, the iDart is the FDA approved ImmunoBlot, and that is a test that's been approved based on blood drawn from a vein. And whether it's through a needle or a port, I don't think that matters at all. The AccuDart is the other one that's now on the market, and that's a finger stick. And that's been cleared based on finger stick. And I don't think it's been validated for blood taken from a port. So I really don't have an answer for that. One other correction I do want to make, the ImmunoBlot is FDA cleared, not approved. It's a matter of terminology. Clearance is for testing, approval is for devices. So if you get like a blood sugar meter, that's a device that gets approval. If it's a test, it gets cleared. So, and just because it's cleared or not approved, it doesn't mean it's only 50-50. No, that's the way the FDA works. So it's an FDA cleared blood test.
Understood. Thank you for that clarification. I'll make sure to keep that in mind in the future. And this one is, I guess, same type of question, except related to the CDC and their kind of standard for testing. Is this woman asked, do you need an ELISA and a Western blot in addition to the iDart? And it's not written here, but my guess would be, in order to have a quote unquote CDC positive or a positive test that is allowing a medical professional to treat you.
Well, actually, that's the whole big deal about this. It is accepted by the CDC when you get the iDart, because the FDA cleared test includes tier 1 and tier 2 right on that ImmunoBlot. So that FDA cleared ImmunoBlot is a 2-tier test, and you don't have to get any other testing.
Amazing. That's very helpful. Thank you.
Yeah, it's a big deal, a real big deal.
So there was one question I see. Hey, David, for how can an individual get this test? Do they have to request it when they go to the doctor or the hospital? Or, how exactly can they get it to be tested?
Well, at this point, anyone can get the Lyme ImmunoBlot from IGeneX. You would get the test kit. You or the physician practitioner would order a test kit from IGeneX and have a phlebotomist draw your blood and fill the tubes, and it gets mailed back to IGeneX. It's a prepaid return mailing box. So that's available right away. In terms of getting a big box lab like Quest or your hospital lab to do it, some hospital and outpatient labs will draw blood and send it to a non-hospital lab, like, A lot of hospitals don't want to have blood sent to a competition. They want to do all their own testing, but some will do you that favor and say, all right, we'll send it out for you as long as you do the mailing, which is no big deal because it includes a mailing kit. But if it's not an agreeable hospital they don't want to send your blood to some competitor, like if you went to Quest, they're not going to take blood out of Quest lab and send it to a competitor like IGeneX. And that's where the FDA clearance is so important. Because now Quest has the ability to buy the kits from iDart, stock it in their own lab so they can draw blood and send it to their own lab for an IGeneX ImmunoBlot. See, that's the real big deal here. You don't have to only use IGeneX once these labs start taking this in as their standard. Now, because it's such a more accurate test, hospitals may actually have two levels of testing. The basic Lyme test, which is their usual not so good IFA, ELISA, Western blot. And they say, well, if you want a better test, we do have the iDart test, the FDA ImmunoBlot. And, that's my hope that they will be doing this. And that's why I think people should start writing to their labs and say, hey, get this kit, stock this kit. It's a much better way of doing it. And because it's FDA cleared, that's federal. That means it's available in New York State.
That's actually a good idea for an advocacy call to action for us as a nonprofit organization is, hey, write, and, we've done a lot of, hey, write a letter to your, your representative to ask for increased Lyme funding. It's, well, hey, write a letter to your local lab to ask for this test to be included there. I think that's a pretty interesting.
And I know there's another very interesting development I've seen. Some big hospital systems, big hospital systems often in competition with each other. In the New York area, there are like several real big hospital systems with many, many branches, and they all want to be in competition. And now, little by little, they're opening their own Lyme disease clinics. And now, if those clinics can offer an IGeneX quality test, as opposed to the standard, IFA that they get from a big box lab, that's going to be a feather in their cap. So if you're a patient going to one of these big hospital-based clinics, let them know, hey, you can get a much better test and you'll beat your competition. So it's going to be actually a sign of pride on their part and a sign of quality to have a Lyme clinic use these better tests. So that's one more bit of action that you guys can all work on.
Agreed. No, that makes a lot of sense, and definitely like the call to action to our patient community to get actively involved in helping moving this forward. I have here, it looks like a couple of questions that overlap in terms of accessibility for this test internationally. I have a couple people from Australia, and I think one or two from Europe that are asking about accessibility of the test.
Well, as I said before, the ImmunoBlot is the one that's cleared is basically the same ImmunoBlot that's been available for many years already from IGeneX. And IGeneX does accept samples from overseas. And I believe they now have some cooperative agreements with European labs. So I don't know the details of this right now, and it'd be up to the lab to talk about it.
Sure.
But, for years, people from overseas have been getting IGeneX testing, so that has not changed.
Awesome. I think this is a good question to end this on, and you may not be able to actually address it, but somebody asked if this new test will potentially have any impact on future treatment protocols as well.
Oh, I think definitely, because now people who have this mystery illness or chronic fatigue syndrome or post something or other can now get accurate testing, and that's been FDA cleared, and no one can say, well, it's a bad test and IGeneX is always positive, don't believe it. No, FDA believes it, so yeah, and the statistics show. So I think it's going to have a huge impact. And we're in the Lyme literate community. We kind of know a lot of this stuff. You go across the street and you find people who don't even know what Lyme disease is still, believe it or not. And they say, well, I'll get a test once a year and see if it's, in my system. I mean, that's the state of poor knowledge out there. So if anybody can go to a local lab or a hospital or even a big box lab and order an IGeneX ImmunoBlot, that's going to change everything.
I agree. I hope everybody who put their questions in feels like we were able to adequately respond. I know that I wasn't able to get to 100% of the questions, but there was a lot of things that overlap. So just really hope that we are able to get to as much as possible. Again, it's being recorded and will be sent out first thing tomorrow morning. So you have an opportunity to rewatch because Dr. Burrascano did cover a majority of your questions in his initial presentation. Thank you so much, Dr. Burrascano, for spending this evening with us and providing this very useful information. It is a huge deal for the Lyme community that this was FDA cleared. and hoping to see future clearances coming out of IGeneX and the FDA here soon. So thank you again.
Yep, and thank you too. And everyone have a good evening.
All right, good night.